Hello students. So we are going to continue with our yesterday's topic. That is, yesterday we started with the reproduction in human being, and we have studied that in human being the reproduction is sexual reproduction, and then we also come to know that the male and female reproductive system are separated. That is, the human beings are showing separate sexes and dimorphism also. That is, you can identify the male and female from external appearance only because of their several changes of body structure as some sort of signs by which you can make out that which is male and who is female. Anyway, we already discussed about the male reproductive system and female reproductive system also. We had discussed by and large. A small portion is left, uh, that will continue now. Okay, so we were here in this paragraph. We have to start. The female external genitalia include mons pubis, labia majora, and labia minora. Now, mons pubis is a fatty deposition which is found on the pubic region, just below the abdomen, which is giving a sort of cushiony appearance. It has no role in the reproduction. Okay, now next is mons pubis. Just below, just after the mons pubis, there are two lip-like folds, or the two folds are found. These folds are called labia majora and labia minora. Labia majora is the outer fold or opening, and just beneath that there is another fold that is called labia minora. Labia majora and labia minora are forming an opening. Okay, labia majora, minora, the two of the labia minora join at the upper end, and just beneath the joining there is a clitoris. There is a small tiny finger-like structure is found, that is called clitoris. The clitoris is the most sensitive part of the female reproductive system, and it gives some sort of pleasure at the time of coitus, and that is a sort of uh, a reason that the people indulge into the sexual reproduction or relations. Anyway, next is the hymen. Hymen is a thin membranous covering which is found covering the what do you call it, cervix, and this is actually it very often it get rupture or damaged when a person or a girl is involved in some sort of activities, sports, gymnasium, certain type of yoga, and horse riding, cycling. So it is mentioned here. See, the hymen is often torn during first coitus. Very often, though, it is related with that. It's certain somehow in our myth it is included as that the coitus should be ruptured during the first intercourse. That gives a sign of virginity. That is, if a girl is virgin, then that when this rupture, it will cause a small bleeding, and that gives a sign that the girl was virgin. But it is not necessary. Nowadays the girls are involved in the so many different type of activities, unlike in the olden time where the girls were very timidly kept, and they were not allowed to do such type of activity. They were not allowed to go out also and involved in such type of activity. But nowadays you see, it can break, it can rupture by sudden jolt or fall, or insertion of vaginal tampon, and active participation in some of the sports like horse riding, cycling. Now, which girl is there who never cycled? Okay, now horse riding is not necessary, but at the same time, long jump, high jump, or gymnastic, or certain type of games, or certain type long, what they call them, stretching and all. So any of these activities, now when a girl is involved in the tennis or badminton, they have to invariably they stretch their legs, and sometimes it may get rupture. Okay, so in such a case, the first coitus will not lead to the any rupture, and it generally give uh, a false information to the boy or a man, or a groom rather, that the girl is not virgin, and it creates unnecessarily problem. And that is why, just for the information, that you should know that there are several reasons by which this can rupture. It is not a valid sign or correct or true sign of virginity. Okay, in some of the women the hymen persists even after coitus. On the other hand, So in such case they had to be, it has to be removed by surgery. So in fact, presence or absence of hymen is not a reliable indicator of virginity or sexual experience. So one must remember this. It has nothing to do with the virginity of a person. It is just a membrane which was found now, but it could be broken or it could be ruptured, and any of those type of activities which we have already mentioned. Okay, now another organ which is found in the female, which is not there in the male, is the mammary glands. And mammary glands in male is highly reduced and it has just a rudimentary structure, whereas in females this is actually part which will be giving nourishment to the newborn child. So a functional mammary gland is characteristic of all female mammals. In all the female mammals, may it be uh, human or a dog or an elephant or a rabbit or a cat or whatever type of mammals you see, they all have this thing. The mammary glands are paired structure. ठीक है? Either it will be one pair as in case of human being, or two pair, four, six, eight, or more. ठीक है? They contain glandular tissue and a variable amount of fat. Okay, so fat is just to give shape. Fat has nothing to do with the this thing, milk production and its discharge and all. So it contains glandular tissue and variable amount of fat. Now glandular tissue of each breast is divided about 15 to 20 mammary lobes containing clusters of cells called alveoli. Alveoli are those bag-like structures in which the milk is produced. Okay? The cells of the alveoli secretes milk, and which is stored in the cavities, that is the lumens of alveoli. We can see lumens of alveoli here. These are fats which are shown in the pink color. Mammary lobes are given here, and these are alveolus. Okay? And mammary duct, and these are lactiferous ducts. So milk comes and filled here, that is in the ampulla and mammary ducts and all. And when this particular part is pressed, the milk oozes out, milk comes out. Okay, so that is what the thing is written here. That is which is covered by the which is stored in the cavity, that is the lumens of alveoli. The alveoli opens into mammary tubules, and tubules of each lobe join to form mammary duct. Several mammary ducts join to form a wider mammary ampulla, which is connected to a lactiferous duct, okay, through which the milk is sucked out. Okay, so with this we have come to the end of the female reproductive system. The female reproductive system has several functions, and those functions are, that is the production of ova, that is secretion of ova, production and secretion of the female reproductive hormone, then the fertilization, then implantation, then gestation, then birth, and then the take care of the newborn, that is by nursing that. Okay. Now the next topic is gametogenesis. So we have studied that is the life of the sexually reproducing organisms that completes in three phases. That is the childhood, or rather it is called juvenile phase, then reproductive and post-reproductive. Juvenile phase itself are divided into three. That is pre-reproductive, reproductive, and post-reproductive. And pre-reproductive are two parts. That is gametogenesis and gamete transfer. Let us discuss all those points. That is gametogenesis, and how gametogenesis takes place in the male and the female. That is how it is there in case of sperm production, spermatogenesis, and ova production, that is oogenesis. So we shall discuss about them one by one. The primary sex organ, testes in male, and the ovaries in female. Primary sex organ, I already mentioned you that primary sex organs are those organs which are actually responsible for production of the gametes. Okay, so male gametes banara कौन, that is testes, and female gametes are produced and released by ova. So these will become primary sex organ. The sperm and ovum respectively by the process called gametogenesis. In testes, immatured male germ cells, that is spermatogonia, produce sperms by spermatogenesis. Let us see, these are the spermatogonia. Thus, spermatogonia cells continually divide by mitosis and add new cells, that we already studied yesterday, so that the total number of the spermatogonia shall remain that many. Otherwise, they will finish off within two or three division, and then there will be no more cells left for the sperm production. Okay, now some of the cells of the spermatogonia, they become mature, and they now known as the primary spermatocyte. The primary spermatocyte is diploid as the spermatogonia cell is. Now the primary spermatocyte will undergo first meiotic division. There are two meiosis, first meiosis 1 and meiosis 2. So first meiotic division, and the first meiotic division, the two cells which are produced in which the number of chromosomes are reduced to half. So from the 46 in human beings, since we are studying the reproduction in human beings, so we are talking about human only. So in human being, the two and number of chromosomes were 46, which are undergone into first meiotic division, and the number of chromosomes may become 23. It is not only the number of chromosomes which are reduced into half, rather they are also developed genetic differences, variations from one another, because during this stage only the crossing over has taken place, and because of the crossing over the variation have already reached there, arise there. Okay, now the second spermatocyte again undergoes second meiotic division and forms spermatids. Okay. Now these see here the 23. I'll just read it, so it will be good for you. That is the prime the spermatogonia, that is present on the inside wall of the seminiferous tubule, multiply by mitotic division. Mitotic division are mentioned here first time. Okay, so mitotic division and increases the number. So that is required. Otherwise, the entire cells will get over within few days. Okay, now each spermatogonium is diploid and contain 46 chromosomes. Spermatogonium. These are spermatogonium. Single is spermatogonium and plural is spermatogonia. Okay, so spermatogonia or spermatogonia which are there, which is mentioned here, that is where it is now? Spermatogonia. Okay, present on inside wall of the seminiferous tubule, multiply by mitotic division and increase in number. Each spermatogonium is diploid and contains 46 chromosomes. Some of the spermatogonia called primary spermatocyte. Some, not all. Periodically undergo meiosis. Okay, not all the time. Periodically undergo meiosis. A primary spermatocyte completes the first meiotic division, that is the reductional division, leading to the formation of two equal haploid cells that are haploid. Okay, तो ये 46 है, ये दो equal cell बनी है, दोनों haploid हो गई हैं. ठीक है, so haploid cells are formed, which have only 23 chromosomes in each. The second spermatocyte undergoes the second meiotic division and produce four equal cells. So four spermatids will form. इनको हमने क्या कह दिया, spermatids. What would be the number of chromosomes in the spermatids? So they will have 1n. ठीक है, haploid. Spermatozoa, that is the sperms, by process called spermatogenesis. After spermatogenesis, the sperm cell become embedded in the Sertoli cells. So here you can see these are the Sertoli cells, and on which the spermatids are just embedded. So this Sertoli cells here serves two important functions. First, they give the nourishment and nutrition to the spermatids, and secondly they also give them shape is important, because if they do not have proper shape, they will not be able to maintain their speed, and if they are not able to reach up to the egg in the proper time, most of them will die. So that is necessary. So the spermatozoa, that is the secondary spermatozoa जो है, once it gets embedded and into Sertoli cell and it mature, and then it separates from there. That separation is called spermiation. Spermiation is a process in which the spermatids get detached themselves from the Sertoli cells. Okay, so that is called spermiation. Now spermatogenesis starts at the age of puberty due to the significant increase in the secretion of gonadotropin. You know gonadotropin, that is the two hormones, that is FSH and LH, releasing, and a gonadotropin releasing hormone is secreted from the hypothalamus, which will induce pituitary, and pituitary will secrete these two hormones, that is FSH and LH, that will induce testes. Okay, so the chain is given here, that is first is hypothalamus. I'll just write here hypothalamus, hypothalamus. Hypothalamus secretes GnRH, gonadotropin releasing hormone, that will work on pituitary, and pituitary will secrete FSH and LH. Okay. Now FSH and LH will go to the testes and where they will act. So the gonadotropin releasing hormone, that is GnRH. This is, if you recall, is a hypothalamic hormone. So we are written hypothalamic. Increased level of GnRH then act on the anterior pituitary gland and stimulate the secretion of two gonadotropin, that is the luteinizing and follicle stimulating hormone. Now let us see what happens to the luteinizing hormone. Luteinizing hormone that is act on the Leydig cells and stimulate the secretion of androgen. Luteinizing hormone. LH क्या करेगा? LH works on Leydig cells to secrete androgens. Okay, androgen कौन सा है? Testosterone. ओके, जबकि एफएसएच क्या करेगा? ये तो ये हो गया, एंड एंड्रोजन इन टर्न स्टिमुलेट द प्रोसेस ऑफ स्पर्मेटोजेनेसिस. ओके, एंड्रोजन क्या करेंगे वो भी लिख देते हैं यहाँ पे. स्टिमुलेट्स प्रोसेस ऑफ स्पर्मेटोजेनेसिस. ओके, I'll just explain how this entire chain works. First, let us study about FSH. FSH acts on the Sertoli cells and stimulates the secretion of some of the factor which help in the spermatogenesis. ठीक है, तो FSH का काम क्या है? केवल spermatid को रिलीज करना है. FSH works on Sertoli cells to release matured spermatids. So these are the function of SPSH and LH. Okay, now let us examine the structure of a sperm. Okay, how a sperm look like, what a sperm look like. So we are already we are right from the childhood we are studying that the sperm is a mobile structure. That is, it's a gamete which can move, whereas the female gametes are always stationary. Okay, so just for the movement it has got its own tail. So if we see the ultrastructure of a sperm, we can clearly see the four distinct organs, that is the head, neck, middle piece and tail. The head is this thing. This is small neck is there, that is the connecting. Okay, this is the middle piece and a tail. The head has got a tip that is filled with a hormone enzyme called uh, hyaluron enzyme, or we can say sperm lysin enzyme. Sperm lysin enzyme is enzyme which is responsible to dissolve the layers of the egg, zona pellucida and corona radiata, and pave the way to get into the new in the egg. Okay, this particular portion from year to year, it is the nucleus, and it has got its haploid nucleus. Okay, and that this particular portion is the nucleus. Nucleus contain one set of the chromosome, that is the gametes are always haploid, so does the nucleus of the sperm also is haploid. Okay, just beneath this portion here you can see a dot-like structure. This is the centromere. So egg has got it, the sperm has got its centromere. That's it. The egg will have its centromere. Okay, and then this is the middle piece. Middle piece has got large number of mitochondria, and these mitochondria will provide energy for the movement of the tails. Okay, so the tail will move and it is vigorously moving, and for the movement it requires ATPs, and the ATP will be produced by these mitochondria. You can see large number of mitochondria which are shown here. These are the mitochondria. Okay, so that is it. And then that's the tail, and the tail will help in the movement. Okay, so sperms will move with the help of their tail. If you read from here, that is, if I read the whole thing from here, that is a plasma membrane enveloping the whole body of the sperm, and the sperm head contain an elongated haploid nucleus. the anterior portion of which is covered by a cap-like structure called acrosome. The acrosome is filled with an enzyme that help in the fertilization of the ovum. How it helps in the fertilization? That is only giving a way to get into the egg. The middle piece possesses numerous mitochondria which produces energy for the movement of the tail. So far whatever we are I told, that is same thing here. Now then, this is important. The human male ejaculate some 200 to 300 million sperms. The number you can have an estimate number 200 to 300 million sperm during a coitus. Now, one thing one must remember, whether coitus has been done or not done, these many sperms are every day produced. Okay, so meiosis is a continuous, mitosis is a continuous process, meiosis is a continuous process to produce the sperms there. Of which for normal fertility, if the fertility has to be maintained, 60% of the sperms should have normal shape and size, and for at least 40% of them should have vigorous motility. So 40% of 200 or 300 million, that is 120 lakhs jo bhi hai. So these many sperms are required. Then only the fertilization. You might have seen number of cases, or in the next chapter we will study, that is there are a number of type of infertility. And one of the most important reason of the infertility is either low sperm count, that is instead of 200 and 300 million, 200 to 300 million sperm, they are maybe producing lesser number, or their motility is less, or their shape is not proper. In any of these cases, they will not be able to. Now, when we are talking about the shape, what does it mean? Shape means that acrosome is not located there. It is located somewhere else. Then how will it take entry into the egg? Okay, so all these things are very important. Now, sperms released from the seminiferous tubules are transported to the accessory ducts. What are the accessory ducts? We already studied in yesterday's class. That is the retitestis is there, vas efferentia is there, epididymis is there, deferens is there. Okay, the secretion of epididymis. Now see, secretion of epididymis, vas deferens, seminal vesicle, and prostate are essential for maturation and motility of the sperm. Epididymis secretes something which will form seminal plasma. Then seminal vesicle will secrete same amount of some amount of fluid. Then prostate will. So these three, and vas deferens is also here mentioned. So they will secrete, and their secretion will give the weight for the sperms to move, and they will make them matured. Okay, now the seminal plasma along with the sperm constitute of semen. Semen is nothing but the fluid medium which contain fructose, which contain calcium ion, which contains some enzymes, and will contain some pH balancing buffering substances plus sperms. Okay. The seminal plasma along with the sperm constitute semen, and the function of the male sex or accessory ducts and the glands are maintained by the testicular hormone, that is androgen. So all these are maintained by the androgen. Androgen, in case of the common name of androgen is testosterone. Okay, let us talk about the oogenesis. Oogenesis markedly different from the spermatogenesis. कोई relation ही नहीं है, except that both of them shows meiosis. Otherwise, there is no similarity present between them. So it's a very often this question is asked, write five difference between the oogenesis and spermatogenesis, and the children write what? That oogenesis take place in the eggs and the or female, and the spermatogenesis take place in the sperm. These are not the thing which has to be given marks. ठीक है, बात तो सही है, लेकिन हम बात पूछ रहे हैं आपसे कि डिफरेंस क्या प्रोसेस के अंदर, आप ये बताओ हो कहाँ रहा है. ठीक है, चलिए let us talk about the differences in their process. The process of the formation of the mature female gamete, that is oogenesis, is markedly different from the spermatogenesis. Oogenesis is initiated during embryonic development stage. Spermatogenesis will begin only when a boy or a child is reached to the adolescence. वो 12, 13, 14, 15, 16, 17 साल का होगा, तब जाकर के उसके अंदर ये प्रोसेस शुरू होंगे. जबकि गर्ल्स के अंदर उसके पैदा होने के पहले, when a girl is only in her embryonic stage, okay, when she is in her embryonic stage, that is if she is on her 25th week of pregnancy, तभी उसमें ये बनने शुरू हो गए. When a couple of million, how many eggs, how many eggs are produced? A couple of million मतलब 2 million 2 मिलियन मतलब 20 लाख. In entire life of a woman, she cannot produce more than 420 eggs in her entire life. Starting from the age of 13, 14, 15, जो भी आप लेना चाहें, maximum age to the 45, 46, 50 ले लीजिए. Then also every month one ova is produced. You can imagine how many months are there from this period to that period. And अगर उसने एक बच्चा भी पैदा किया तो there will be a phase of a phase in which the ovulation will stop, the oogenesis will stop. So वो कम से कम 9 महीने तो रुकेंगे, 10, 11, 12 महीने तक, 6 महीने मतलब हो सकता है 15, 16 महीने तक तो ओव्यूलेशन हो गई ही नहीं. So then the number of sperms eggs produced are all the more decreased. So what I mean to say is that is the number of that is the female gametes जो produce होने हैं total, उसके comparison में यहाँ पे कितने बन गए हैं, that is there are couple of million, that is 20 million gamete mother cell, that is oogonia. Sperm mother cell को हमने कहा था spermatogonia, इनको कह रहे हैं oogonia, are formed in each ovary. तो अब ये भी देखिए, 20 लाख एक ओवरी में, लेफ्ट वाली में 20 लाख राइट वाली में, तो 40 लाख हो गए फॉर्म्ड विद इन ईच फीटल ओवरी. नो मोर oogonia. अब इसके बाद क्या ऐड करना है? बट हम स्पर्म में क्या पढ़ रहे थे? द स्पर्मेटोगोनिया आर कंटिन्यूसली एडेड बाय माइटोसिस. हियर कोई माइटोसिस नहीं है, हो गया जो होना था हो गया, नो मोर विल बी एडेड. ठीक है, तो अगेन देयर इज अ डिफरेंस, दैट इज नो मोर oogonia आर फॉर्म्ड एंड एडेड आफ्टर बर्थ. जो होना था वो हो गया, अब और कुछ नहीं ऐड होंगे, but rather their number will decrease. These cells start division and enter into the prophase 1 of meiotic division and get temporarily arrested at this stage called primary oocyte. ठीक है, ये जो सेल्स हैं अब ये कहाँ पर आकर के रुक गई? प्राइमरी oocyte, तो मतलब फर्स्ट मियोटिक डिवीजन का प्रोफेज 1 पे आके अटक गई. In spermatogenesis, we have studied the primary spermatogonia undergoes two meiosis continuously, one after the other. First meiotic division and second meiotic division. First meiotic division, number of chromosomes are reduced to half, and second they are maintained to the same. Okay, यहाँ क्या पढ़ रहे हैं? Division तो पूरा हुआ ही नहीं है. फर्स्ट म्यूटिक डिवीजन दे जस्ट एंटर्ड एंड वो क्रॉसिंग वगैरह जो कुछ होना था वो हुआ, हो करके एंड वो वहीं पर रुक गया है, सस्पेंडेड. ओके, तो जितने भी हैं वो सब वहां पे आकर के सस्पेंड होना. ईच प्राइमरी oocyte then surrounded by a layer of granulosa and then and then called primary follicle. तो जब ग्रेनुलोसा बन जाएंगे तो उनको क्या कहते हैं? प्राइम. A large number of these granulosa degenerate during the phase of the birth to puberty. Very important. 40 लाख थे ना, 20 लाख इन ईच ओवरी. बनाने तो ही नहीं इतने, बनाने कितने? 420. मतलब फ्रॉम ईच ओवरी मे बी 210, 230, 240, 300 मान लो जी, लेकिन बने तो इतने सारे हैं. तो अभी क्या है इनका? धीरे 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 डिजनरेशन. बोले वाई अननेसेसरली स्पेंडिंग एनर्जी, वाई यू आर ईटिंग आवर फूड, वाई यू making us trouble. So the number of chromosomes, this oogonia will start decreasing. Therefore, at puberty 60,000 to 80,000 primary follicles are left in each. अभी भी काफी ज्यादा है. 60,000 टू 80,000. 80,000 तो दोनों ओवरी में मिला करके अभी भी 1,60,000 हो गए. ठीक है, 1.6 मिलियन, 1,60,000. हाँ, ठीक है, तो ये तो काफी मतलब काफी ज्यादा है. 16, 1,60,000. हाँ, तो 1.16 मिलियन. तो इतने सारे रह गए, अब बस बाकी सब खत्म हो गए ना. Primary follicle gets surrounded by more layers of granulosa cells and a new theca called secondary follicle is formed in that. Okay, the secondary follicle soon transform into a tertiary follicle which is characterized by a fluid-filled cavity called antrum. अब ये एंट्रम और ये सब देखने के लिए हम यहाँ पर आ जाते हैं. ये प्राइमरी फॉलिकल्स हैं. सी, ये छोटी छोटी स्टेजेस. प्राइमरी फॉलिकल जो है ग्रेनुलोसा लेयर एंड ये सब बन करके थोड़ी और बड़ी होगी. Then an antrum cavity is formed. This is a tertiary follicle with a large antrum. That will all the more grow. ग्रेफिन फॉलिकल बन जाएंगे. ये मेंस्ट्रुअल साइकिल की फेजेस के साथ में चेंज हो रहा है. ओके, एवरी मंथ वन, आइदर फ्रॉम लेफ्ट और फ्रॉम राइट. तो ये फेजेस के अंदर होंगे। एक इस महीने मैच्योर हो रहा होगा, दूसरा जो इसके पीछे वाली वो अगले महीने, उससे पीछे वाली और उसके अगले महीने, एक एक महीना लग जाता है इसको मैच्योर होने। ठीक है, तो देखिए द थीका लेयर ऑफ द ऑर्गेनाइज्ड इनटू इनर थीका इंटरना एंड आउटर थीका एक्सटर्ना, एंड इट इज इंपॉर्टेंट टू ड्रॉ योर अटेंशन दैट एट दिस स्टेज प्राइमरी oocyte within the tertiary follicle grows in size and completes its first meiotic division। फर्स्ट मियोटिक डिवीजन टर्शरी oocyte बनने के बाद, प्राइमरी oocyte से लेकर के जो है टर्शरी फॉलिकल तक आते आते उसका फर्स्ट मियोटिक डिवीजन कंप्लीट। सेकेंड मियोटिक डिवीजन विल कंप्लीट ओनली आफ्टर फर्टिलाइजेशन। हुआ तो ठीक है, नहीं हुआ तो कोई जरूरत ही नहीं है इसकी। So it is an unequal division resulting the formation of a large haploid secondary oocyte and a tiny polar body। Again it's a difference, we never studied in this form that two cells will be unequally, एक बड़ा होगा एक छोटा होगा, no 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 no। In males we have studied one primary spermatogonia will develop into two secondary spermatogonia will develop into four spermatids, and four spermatids will form four sperms। ठीक है, देखिए यहाँ क्या पढ़ रहे हैं, जो वन प्राइमरी क्या कहते हैं oocyte थी, दैट प्राइमरी oocyte इज ग्रोन, एंट्रम इज फिल्ड, सेकेंडरी एंड टर्शियरी स्टेजेस आर फॉर्म, एंड फॉलिकल के स्टेजेस आई हैं। उसके बाद क्या होगा, डिवीजन होगा। डिवीजन होने के बाद क्या है, दिस डिवीजन इज अनइक्वल डिवीजन, वन विल फॉर्म सेकेंडरी oocyte एंड द अदर विल फॉर्म पोलर बॉडी। द सेकेंडरी oocyte इज लार्ज, पोलर बॉडी वाज सो टाइनी कि उसका कोई अस्तित्व ही नहीं है। And आगे लिखा है देखिए, it is an unequal division resulting the formation of a large haploid secondary oocyte and a tiny polar body। The secondary oocyte retain bulk of the nutrient rich cytoplasm and the primary oocyte। ठीक है, एडवांटेज क्या है कि भैया वही तो काम आएगा फॉर द टाइम ऑफ फर्टिलाइजेशन टू its entry into uterus and implantation। Whatever energy is required, that much of food is there। Does the first polar body born out of the first meiotic division divide further or degenerate? We are not certain about। कम से कम इस बुक में तो आपको एग्जाम में आता आंसर यही लिखना, we don't know anything about polar bodies। Okay, at present we are not very certain about this। The tertiary follicle further changes into mature follicle and or Graafian follicle, and the secondary oocyte form a new membrane called zona pellucida। अब इसने अपने चारों तरफ एक लेयर बना ली, इसको कहेंगे जोना पेलोजिडा। द ग्राफियन फॉलिकल नाउ रप्चर्स ऑन द रिलीज ऑफ oocyte। In the mid of the menstrual cycle, there will be some changes taking place in the hormonal level, that is LH, luteinizing hormone. And under the influence of luteinizing hormone, the matured secondary oocyte will be released. And this process is called ovulation। अभी कितने अंडे निकलते हैं एक महीने में? ओनली वन एग। So how many this primary oocyte should develop into secondary, tertiary and other Graafian and follicle वगैरह सब में कितने? That is in a month one, either from left or from right। So now you can differentiate। यहाँ पे आपको लिए difference है। This chart is very important। Let us talk about this। तो यहाँ पे दिया spermatogenesis, दिया oogenesis। Spermatogenesis कब शुरू हुआ? At puberty। ये कब शुरू हो रहा है? When in the fetal life। Here it is mentioned fetal life। ठीक है, let us finish first spermatogenesis। The spermatogonia, primary spermatogonia, then it will divide by the mitosis। बना काफी सारे बना दिया उन्होंने। उसके बाद ये प्राइमरी स्पर्मेटोसाइट है, दैट विल अंडरगो फर्स्ट म्यूटिक डिवीजन, इक्वल है, दो स्पर्मेटिड बन गए। सेकेंडरी स्पर्मेटिड जो है दैट विल अगेन अंडरगो सेकेंड म्यूटिक डिवीजन, दैट विल फॉर्म फोर स्पर्मेटोगोनिया। ये फोर स्पर्मेटोगोनिया बन गए। फोर स्पर्मेटोगोनिया विल फॉर्म द फोर स्पर्मेटिड्स, स्पर्म्स। ठीक है, जिनको हम स्पर्मेटोजोआ कह देंगे। वेरी सिंपल। कब शुरू हुआ? जवानी आने पे। गर्ल्स की बात करते हैं। द oogonia formed couple of million कब बने? इन द एम्ब्रियोनिक स्टेज, फीटल लाइफ। ठीक है, माइटोसिस डिफरेंशिएशन जब होना तो हो गया। Once a child, a girl is born, afterwards no more addition, no more mitosis, whatever made are made। So उनको हम कह देंगे प्राइमरी oocyte। Now primary oocyte will undergo first meiotic division prior to ovulation. Once she will reach to the puberty. Here written it is. Birth, उसके बाद childhood, then puberty. प्यूबर्टी तक फर्स्ट डिवीजन पूरा नहीं हो रहा, फर्स्ट मियोटिक डिवीजन अटक गया जा करके प्रोफेज 1 में. नाउ इट विल कंप्लीट ओनली इन द प्यूबर्टी एंड दैट आल्सो वन इन एवरी मंथ. ठीक है. और उसमें टू सेल्स विल फॉर्म, वन द प्राइमरी बॉडी एंड द सेकेंडरी oocyte. The secondary oocyte which is formed will again go another meiotic division but only आफ्टर फर्टिलाइजेशन. तो ये लिखा एडल्ट रिप्रोडक्टिव लाइफ एंड उसके बाद यहाँ पे आकर के तो ये रिप्रोडक्टिव लाइफ जब हो जाएंगे दैट इज व्हेन she'll मेट विद द ऑपोजिट सेक्स एंड उसके बाद में उसका गैमेट का फर्टिलाइजेशन. आफ्टर द फर्टिलाइजेशन सेकंड मेयोटिक डिवीजन कंप्लीट. सो प्लीज रिमेंबर दिस इसको अच्छे से रट लीजिए, अच्छे से घोट लीजिए. सो the important parts we already studied. In tomorrow's class we'll study about menstrual cycle and then how the further development take place. So that's all children. Good day, enjoy your day. Thank you.